Now we are moving on to Chapter 3. We've covered Chapter 2, which is basic chemistry, so this will lead us directly into some organic chemistry that is more along the lines of the chemistry of life. Organic chemistry is basically the chemistry of carbon-based molecules. This is a different definition of organic than maybe one that you're used to. Organic food obviously refers to food grown without pesticides or other chemicals. Organic chemistry doesn't have anything to do with that. It's more the chemistry of carbon. We're going to look at carbon chains, rings, and the functional groups attached to them. These functional groups basically determine the reactivity of the molecule. Carbohydrates are made of long strings of carbon or carbon rings. And eventually we'll start talking about proteins, their function and structure, and DNA and RNA, because those are also carbon-based. Those are the molecules that guide our lives. Let's go a little deeper into talking about the chemistry of carbon. As we've mentioned many times, carbon has 6 protons, 6 neutrons, and 6 electrons. We've talked about the shells. You know where the electrons are, inner and outer, and why carbon makes 4 bonds. Carbon nearly always makes 4 bonds. It's very happy doing so. And carbon will most frequently be found in chains or rings, as you're going to see in some of the following pictures. Functional groups provide reactivity for these carbon chains and rings. When carbon makes its 4 bonds, it's pretty happy. Like methane we saw before. It's not looking to do anything else, but if it's connected to some other atoms or molecules, it will react. These 2 pictures I want to show you to give you an idea of how electrons circulate around carbon and just grasp what carbon looks like. Here's a diagram like we have in the book showing your protons, your neutrons, and electrons, the inner shell and the outer shell. This is important to understand, how the bonds happen, why they always happen, and 4, we've already covered that. I included this picture so you can get maybe a little better idea of how the electrons circulate the nucleus. These are the outer shells here, and then the inner shell. Okay, we're going to start by looking at diagrams of molecules that you'll see in your book, or how you're going to write them if you ever have to write them down. These are called skeleton structures. These are chains, carbon chains, saturated carbon chains, because every bond that carbon can make is full, and carbon is connected to a hydrogen. As you can probably see, if carbon is connected to another carbon, there's not a hydrogen there, but it's still a single bond. These chains can get very, very long. A symbol like this represents a double bond, and I'll show you later what that looks like in a line drawing. They may be branched, and the branches you can see right here, this will change the name of a molecule if it's branched as opposed to in a line. What you're going to want to become more familiar with is carbon rings. This is an extremely strong and stable arrangement for the carbon atoms, especially when you've got the double bonds here and then a single hydrogen off each available carbon. If you'll look, carbon has 4 bonds at each location. This structure is very, very strong, and this is one of the most common structures in biological or organic molecules. So another part I want to mention, now this here, the book talks about monomers and polymers. A monomer, see, would just be one little carbon connected to a bunch of hydrogens, not both. This is a polymer because there are more than one. That's something I want to keep in mind. Now these structures, if I draw them on the board or if you see them in your book, these will be shown as zigzag lines. If they're just single bonds between carbon, they're a zigzag. When there's a double bond, it'll be a straight line, and the rest of this will be a zigzag. You'll see that in this slide. In a picture like this, there's a single carbon at each peak on the zigzag. And where you see that double line, like between the numbers 9 and 10 on the top picture, that's a double bond between 2 carbons. If that top picture were all zigzags without the double bond in it, that would be saturated. So what's attached to these polymers or these carbon chains would be functional groups. The functional groups determine the reactivity. There are many functional groups, but the two that we're going to talk about most are carboxyl and hydroxyl groups. Carboxyl groups are where the C at the end of the carbon chain has a double bond to an O and then a single bond to a hydroxide ion. And then the bottom picture shows you the hydroxyl group, where that end carbon is connected with a single bond to an O, which is connected to a hydrogen. The R represents a carbon chain there. That's just a shorthand version of saying there are carbons attached here. It could be attached to anything. It's just a way to show the functional group without having to show the entire picture of a molecule. Now these two will work together. Most frequently, an OH will pop off and an H. They come off together, which is called dehydration, and then these two would bond or connect, well, to form a larger molecule. So when that dehydration reaction occurs, that's how we get long chains of carbon or carbon rings, and we call those carbohydrates. This is what your body breaks down into sugar to gain energy. So essentially carbohydrates are sugar, and you'll see the strings of them here in just a moment. The ring structure is of course one of the most common shapes for carbon to take. What I want to show you here, so sugar is saccharide. Up at the top, this is a monosaccharide with an OH. This is also a monosaccharide. Glucose and galactose. When you have the dehydration reaction like we talked about, this hydrogen's connected to the oxygen. This is obviously connected to the carbon. H2O is right here. That's a very happy molecule. When this reaction occurs, hydrogen comes off. This is called dehydration because you're removing water from the overall system. The bond is made here. Remember, see, this oxygen becomes this oxygen, and you get a disaccharide, 2 carbon rings, called lactose. Now that's just the beginning of showing you what a carbohydrate is made of. As you know, you don't use absolutely everything that you eat. Your body has to store some of it because there are times when you may not have food available. The way potatoes do so, or a plant, is they store that energy as starch. And looking at this picture, you can see all these little green things are 6 carbon rings. And this is the way a potato will store its sugar. This is also what we would eat to gain energy from it. Now, our body will store it as well, or use it as building blocks to build our own tissue. This picture shows, I love this picture because it's got everything in it and it's wonderful. These are your muscle fibers. You would turn starch or glucose into glycogen as a form of storage. As an animal, that's what we do. So it looks extremely similar. You see the oxygens in the same places, but it's a branched chain. Perhaps it's a little stronger, a little tougher. Obviously when you bite a potato, it's a little easier to bite than, say, biting into muscle tissue. Or you can look at the way plants form their tissues using stored sugar in a more linear fashion to create a strong fiber, and they use hydrogen bonding to keep them in order. In order to understand how these large molecules work, we need to understand the difference between hydrophilic and hydrophobic. I know you've heard these base terms before, where if something is philic, it loves it. Phobic means it fears it, essentially. I want to show you fructose here because it's a highly hydrophilic molecule. This is where you're going to need to incorporate your understanding of electronegativity of something like the water molecule, where it's more negative on one side and more positive on another. That's what's happening in this fructose molecule as well. See all the hydroxide is here? Oxygen, remember, has a very strong pull on electrons, so it's going to hold on to the electrons that it shares with hydrogen and carbon just a little bit longer than the other 2 atoms get to. And that's happening. See, now this is a 5-part ring with oxygen at the top, so we've got carbon, 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 and carbon here, with an additional functional group off of the edge. Because this hydroxide is a little further away than the main group, it will have a stronger negative moment around this oxygen than, say, this oxygen, because it's closer to many other carbons. But essentially, that makes, with all of the oxygens attached to this particular molecule, it likes water. It will dissolve in water. It will connect to water readily. For the hydrophobic molecules, this is your glycerol component. This is what the basic structure of all fatty acids. Then you have a functional group right here and a hydrocarbon chain. This one's saturated, this one's saturated, this one is not. You see it's got a double bond in it, so it's got a different structure. This is also called a triglyceride. 1, 2, 3, connected to a glycerol, triglyceride. These are lipids, these are fats, these are oils. You can tell they're hydrophobic because oil floats on water. It's less dense, but it will not mix with water, as you've seen. The other thing to keep in mind, like we talked about, oxygen is strongly electronegative, so it holds on to those electrons, making it more polar. Carbon and hydrogen do not do that. It is not polar. It's completely happy. Everything is equally shared among all the bonds. When all three fatty acid chains line up, that particular substance will become more solid, say at room temperature. That's why things are hydrogenated. If it's an oil at room temperature and some company wants to make, something more like butter so it's harder at room temperature, they would hydrogenate it. Okay, as we're talking about hydrophobic and hydrophilic, I just want to throw in something here about how soap works. The molecules of soap, on this chart they call it an anion of a fatty acid. Basically, it's a large molecule where one end is very polar. It has, like in this particular picture, a negative charge on it, and the other end of a soap molecule is not polar. So one end is hydrophilic, one end is hydrophobic, and this is how soap makes bubbles. You'll be able to see that in this picture here. The dark squiggly lines are fats. The red ones are soap, and these are water molecules. They also show cations, but I don't want to confuse you guys, so I'm not going to talk about that. But as you can tell right here, the way the soap molecules orient themselves is such that the polar or charged end is oriented outwards with the water because it's hydrophilic, and then the hydrophobic tail is focused inward where there are other like molecules, like fat essentially. So this is how you can use soap to wash something away. The molecules essentially surround the fat part and can be washed away because they will attach to water, sort of like hydrogen bonding. We've already talked a little bit, well, enough about what something means to be hydrogenated. It is an artificial process where hydrogen gas is bubbled through a substance with a precious metal catalyst to essentially make it more solid or change the bonds on the fatty acid chain from any double bonds or oxygens or whatever else is in there, to replace that so that each fatty acid chain is a solid hydrocarbon, just carbon and hydrogen all the way down. When that happens, the way a substance can become more solid is because it is then able to stack more closely. Basically, the molecules can become closer and the substance itself can perform more like a solid as opposed to an oil at room temperature. We've talked about sugar and carbohydrates, so other organic molecules that are important in your life are steroids, which are also hormones, carbon-based, rings and chains and other structures. Now, steroids are where these molecules become more complicated. They're not in strings as clearly as carbohydrates, muscle fiber, proteins. Well, proteins are complicated. We're going to get into that in just a moment. Just so you know, steroids are used in chemical signaling in the body, hormones. What I know most about steroids are how pollution, when it enters a living body, can mimic a steroid and cause your body to react as if it were a steroid or hormone. I've included a link here since the PowerPoint is on the website. I'd like you to check it out if you're interested. Moving from steroids directly into proteins. Proteins, well, sort of like DNA has 4 building blocks, proteins have 20 basic building blocks, and the basic building block of a protein is called an amino acid. Amino refers to nitrogen. Anything in chemistry, if it's an amine or amino, it's got some level of nitrogen in it. You are made of proteins. Most things are made of proteins. Here's the basic structure. Here's your amino group, nitrogen. your carboxyl group over here on the edge, your monomer carbon in the middle, although it's not a monomer anymore because it is connected to the nitrogen and the carbon. It's got 4 bonds, it's got 4 bonds, everybody's happy. And then your side group. This is what is used to determine what the amino acid is called. Here it's leucine. There are 4 carbons attached. This is hydrophobic, it's all carbons and hydrogens. The amino acid serine. Because it's got the hydroxide attached, it's hydrophilic. Different amino acids are either hydrophobic or hydrophilic. These cause the long protein chains to fold into very specific shapes, determining what they can do and what services, I'm going to call it services, what services they can perform in your body. When amino acids are bonded together, this is the same reaction that we've talked about before, where it's a dehydration reaction. Water is taken off. You have a hydroxide and a hydrogen atom here that can bond, which will leave this nitrogen and this carbon to create a covalent bond. The bond between amino acids is called a peptide bond. That's just what you need to know so that you know what we're referring to when we talk about it. When you have many amino acid groups linked together from their basic backbone here, it's a polypeptide. Remember, poly means many. So moving on to talk more about protein shape, because these proteins are made, proteins are made of very, very, very long chains of amino acids. This is a representation of the chain that you will get. This is simply the chain part. The hydrophilic and hydrophobic side groups will help determine the secondary structure of a protein. A protein is a very, very complex folded molecule. It's not as simple as DNA, where it's just a double helix. This is your string. This is the primary structure. This is the language or the code of the protein. Based on the side groups, it'll make a secondary structure, which may be pleated. This is hydrogen bonding here. Or it may be a spiral with hydrogen bonding here. Now this can be different along the chain. Some of it may be pleated, some of it may be spiraled. That's when we move forward into the tertiary structure of the protein, where all these folds and pleats then come together again to form a different shape. This is a polypeptide section. The whole thing is a polypeptide. If you can tell, it's like a ribbon all coiled up here. The tertiary structure is generally the final structure of a string that makes a protein. Not all proteins, but some. As you can tell, if there's more than one string attached to an initial base here, then it'll have a quaternary structure, where each string of amino acids will come together to form the ultimate size and shape of the protein. Now, size and shape of proteins is absolutely critical, because these proteins are shaped very specifically to, say, fit around another molecule that they are designed to break the bond of, like glucose. That's how you get energy out of the sugars that you eat. But if there is a mistake in the chain, it will not form the right shape, and it will then not be able to perform the function it's designed to do. I just want to show you the final structure of a protein so you can see what I'm talking about. This is extremely complicated. This one may not have a quaternary structure. This may just be the tertiary structure, one long string of amino acids making a protein. And this will be the target. Let's call it fructose, or, I'm sorry, lactose. That little red target fits very specifically into the opening of that protein. And the functional groups, or the side groups, on the amino acids are positioned just so the particular reactivity will break that little red target apart. One more thing I want to mention about the functional groups is that, like we've talked about, if it's distorted, they will no longer work anymore. Your book has an excellent example of sickle cell anemia, where one slight change in the amino acid chain that makes a hemoglobin cell will make it such that in a particular environment it changes shape into more of a sickle form as opposed to the round form, and it can no longer then carry oxygen to the rest of the body. That's pretty serious, and it's just, I believe, one amino acid difference. One little amino acid can make that much of a difference. So that's why we are incredibly intricate, detailed, complex creatures. DNA and RNA. You know about DNA. That is the blueprint for life. But how is it a blueprint for your life? We'll talk about that in a moment. So I want you to know that DNA is deoxyribonucleic acid. RNA is ribonucleic acid. And the connection between the two is that DNA is a code that other proteins read to make RNA. RNA is then another code that is read to form an amino acid chain, which is a protein. And in the picture, the blue double helix is the DNA, the red is the RNA, and the purple dots are the amino acids. So this is the base structure of a DNA nucleotide. Thymine is essentially the nitrogenous base or the functional group. It could be A, C, G, or T, adenine, guanine, thymine, or thymine. But all DNA has this phosphate group and a sugar, deoxyribose. And this is the backbone. This makes the backbone of the double helix. Just to show you a little bit, phosphate group, sugar group, base. Now the difference between DNA and RNA. DNA's nitrogenous bases, or functional groups, are A, C, G, and T. RNA are A, C, G, and U. It's got uracil instead of thymine. The base sugar is different. Deoxyribose. Ribose. So deoxy means, you see how there's no hydroxide ion here? Here there is. That's the main difference. And then DNA forms a double helix, where RNA forms a single-stranded spiral. What we'll talk about more later in class is how DNA is read by proteins within the nucleus to create an RNA strand, which is one of the few things that can be formed and taken outside of the nucleus. It's like a lockdown secure zone where the proteins are made in the cytoplasm of the cell, and then they can be transported in or out of the cell for other functions that they need to perform. We have covered the chemistry of carbon, carbon chains, rings, and functional groups, the structure and breaking down of carbohydrates, the importance of proteins, their function and structure, as well as the function and structure of DNA and RNA.